All right, we're going to do the same we've been doing for reviews, looking at the cup, the Kahoot, um, and then going over everything. So the function of the upper respiratory tract is for gas exchange, moisten and filter air, bronchoconstriction and bronchodilation, or produce surfactant. So the upper respiratory tract is moistening and filtering air. So we have nasal hairs, we have the concave, we have the meatus, going through all of that, adding humidity and filtering, make sure we're getting any dust or dirt out of the air. True or false, pneumocytes type 2 produce surfactant. This is true. That's the structure. Again, pneumocytes type 2 produce surfactant. That's to make sure that we keep the alveoli open, the lipid kind of solution that repels water. So we have water droplets that will not collapse the alveoli, keep it open. Pneumocytes type 1 are going to be the squamous cells that are allowing for gas exchange. If you increase volume, pressure decreases. What law is this? Boyle's, Henry, Dalton, Fox. This is Boyle's law, so I'm gonna switch screens. Go this. So again, remember Boyle's law is if pressure increases or if space or volume increases. So when you breathe in, you are increasing your thoracic cavity. So there's more space. So the air can spread out more. So the pressure is actually going to decrease. That allows, again, pulmonary ventilation because there's less pressure. So you go from high pressure outside the body to low pressure inside the lungs because we've expanded and increased that volume. Then with the elasticity or the recoil of the lungs, we decrease the volume to get smaller, so there's more pressure. Because same kind of the air is going to bounce off the walls and hit the walls more often. There's more opportunity for it to kind of be compact. Again, think of it as a big room. So you have 20 people in an auditorium. They have a huge room. Well, they can spread out and they can walk blindly and not hit the walls as often. Take that same 20 people and stuff them in a teeny tiny room. So if they're walking around blindly, they're hitting the walls, they're hitting each other. So the pressure is going to increase. So think of it that way. Back to an increase in pressure outside a liquid causes an increase in the amount of gas that goes into solution is what law? That's Henry's law. So, go back to my PowerPoint. So again, we look at it. So this is again for looking at gas exchange, external respiration. So you have fluid and you have the air above it. So if you're increasing the pressure, then you're going to force more air into the solution, into the water, versus if you're decreasing the pressure, there's going to be less. And you can think of it just kind of more, even with Boyle's law, if you're decreasing the space and you're increasing the pressure, then more is going to go into solution versus less. If you decrease the pH, what happens to the hemoglobin saturation curve? It shifts to the left, stays the same, shifts to the right, increases. So this shifts to the right. Uh, So remember the saturation curve kind of looks like the S where it comes up and it's getting closer to 100, and then you can kind of affect it. So the percentage of hemoglobin that's going to be have all of the oxygen on it is going to get close to 100 as partial pressure of oxygen. But if you change the pH, you're going to shift it to the right. So it's going to be different because you've increased pH or decreased pH. What's going to happen is if you are decreasing pH, you want to decrease the affinity of oxygen to stay with hemoglobin. You don't want it to stay with hemoglobin. You want it to leave because if there's a decreased pH, most often there's increased metabolic activity. So you want more oxygen to the tissues. So think about it that way. If you are shifting the curve to the right, then you are decreasing its affinity to stay with hemoglobin and increasing it in the tissues. So all of the things that you think of, you want more oxygen in the tissues, then it's going to shift the curve to the right. A decrease in temperature means hemoglobin will release oxygen more readily. True or false? This is false. This would be true if it said an increase in temperature. Because remember, an increase in temperature is meaning you're working your muscles more. You want to have hemoglobin give up oxygen more readily because you want it in your muscles and your active tissues. The epithelium of the alveoli is what type? Simple columnar, ciliated, pseudostratified, simple columnar with microvilli, or simple squamous. So this is going to be simple squamous. Remember, those are the flat football kind of looking ones. They're going to have the smallest distance for diffusion, and that's what we want because the epithelium or the pneumocytes type 1 are where diffusion's occurring. Oxygen's going into the body and carbon dioxide's leaving. Pulmonary ventilation is exchange of gas between the alveoli and pulmonary capillaries. True or false? This is false. Pulmonary ventilation is air going into the lungs and out of the lungs. This is the amount of air left in the lungs during a respiratory cycle. So this is residual volume. So there is some air that stays in the lungs to keep all the alveoli open, and kind of stays inside the lungs. Tidal volume is the normal kind of breathing in and out. Inspiratory reserve volume is how much more past the normal breath you can inspire or breathe in. Expiratory reserve volume is anytime past normal that you can push air out or expire. Hypercapnia is an increased amount of carbon dioxide, true or false? This is true. Hypocapnia is a decreased amount of carbon dioxide. Hypercapnia is an increased amount. So this is going to be that you are not breathing as much because you're keeping carbon dioxide in. Hypocapnia is breathing too much and you're pushing out too much carbon dioxide. This specific part of the brain stimulates inspiration by stimulating respiratory muscles. Um, Ventral respiratory group, dorsal respiratory group, apneustic center, pneumotastic center. This is the dorsal respiratory group in the medulla. It's innervating the diaphragm, and mainly and going to cause it to contract and increase the space in the thorax so air will come in. That's actually oxygen levels are the most important stimulation of breathing. That should be that. This is false, kind of give you the answer. The most important stimulation for breathing is actually carbon dioxide levels, not oxygen levels. What is a possible side effect of weakening the lower esophageal sphincter? Uncontrollable bowel movements, heart attack, acid reflux in the esophagus, or rapid overfilling of the stomach? Remember, the lower esophageal sphincter is synonymous with the diaphragmatic constriction. So this is going to be the lower or inferior part of the esophagus. If that is open too much, then there can be acid or the hydrochloric acid that's going into the esophagus and causing issues. So that's going to cause gastroesophageal reflux disease, which is painful. Can cause ulcers because hydrochloric acid shouldn't be in the esophagus. Only the stomach is made to handle it. Peristalsis is part of which digestive process? Absorption, ingestion, propulsion, or chemical digestion? So this is moving stuff along the digestive tract. So remember, peristalsis is contraction of the smooth muscle in a rhythmic way and relaxation to push substances through the GI tract. So the parietal cells are responsible for hydrochloric acid production and intrinsic factor. Chief cells, remember, are pepsinogen that are released, and it needs the hydrochloric acid from the parietal cells to convert to pepsin. So they kind of work hand in hand. And G cells, D cells are more hormonal. The membrane that lines the body wall of the abdominal cavity is ventral mesentery, visceral peritoneum, parietal peritoneum, or retroperitoneum. So this is the parietal peritoneum. Both are going to be peritoneum, but this visceral peritoneum would be along the organs, like the of the specific areas of the digestive system. Parietal peritoneum is on the body wall. Retroperitoneal is what we call if any organs are behind it or not outside of the two layers of peritoneum. And mesentery, remember, is between the small intestine. Excessive acidity in the stomach causes a blank and blank secretion. So decrease in stomach acidity or excessive acidity, sorry, is in the stomach is going to cause a decrease in gastrin because gastrin is going to stimulate acid release in the stomach. So you want less of it around if there's already too much acidity. So you don't want more released if it's excessive already. Now I kind of give this away, but the gastric cells that secrete pepsinogen are these are the chief cells. Remember, chief cells are secreting pepsinogen, the inactive form of the protein digesting enzyme, and then converted to pepsin by the parietal cells with their hydrochloric acid. Most of the digestive enzymes found in the small intestine are secreted by this accessory organ: liver, gallbladder, stomach, pancreas. So this is secreted by the pancreas, all those digestive enzymes, or most of them. This substance secreted by the pancreas helps neutralize chyme, chymotrypsin, bile, trypsin, bicarbonate. Remember, chymotrypsin and trypsin are protein digesting enzymes. Bile is for fat emulsification and digestion. Bicarbonate is a buffer, so that's going to increase the acidity of chyme, especially in the duodenum. Amylase allows us to digest carbohydrates, proteins, fats, all of the above. Remember, amylase is for carbohydrate digestion. Remember, there's salivary amylase, so it starts in the mouth for carb digestion, and then we have pancreatic amylase that's going to continue to digest carbohydrates. Why is the small intestine well adapted for nutrient absorption? Acid secretions from the stomach are neutralized in the duodenum. CCK is released from the duodenum to promote liver and pancreatic secretion. Large surface area due to villi and microvilli. Small intestine is very long and thin. So there's tons of surface area in the intestine, small intestine, with the villi that you kind of on the surface, and then the microvilli on the surface of the cells. So there's tons of surface area that allows plenty of space for nutrient absorption. The functions of the large intestine include all of the following except elimination of waste, absorption of water and electrolytes, absorption of nutrients, absorption of vitamins produced by the bacteria. So this is absorption of nutrients. So the large intestine doesn't do this. It gets rid of waste. It absorbs water and electrolytes that are still there, and it'll absorb any vitamins that are produced by the bacteria. But all the nutrients that are absorbed have been absorbed in the small intestine. These cells release intrinsic factor, chief cells, parietal cells, G cells, D cells. These are parietal cells, so along with hydrochloric acid, parietal cells release intrinsic factor, which is essential for vitamin B12 absorption. So to absorb vitamin B12, we need intrinsic factor. Which of the following reflexes are we able to voluntarily control? Gastroileal reflex, gastrocolic reflex, enterogastric reflex, or the defecation reflex? So we're able to control defecation for the most part. Gastroileal reflex, gastrocolic, and enterogastric, all foods hitting the stomach, and there's going to be signals going to the ileum to kind of empty out into the colon to empty out as well, and then the small intestine to start getting ready for food because it's going to be coming out of the stomach eventually. Movement in the large intestine is due to small haustral contractions, true or false? This is false. The haustra do contract. There is contraction there, but the major movement in the large intestine is due to the mass movements that happen 3 to 4 times a day, where there's just a major movement of the large intestine all along to push feces into the rectum. True or false? Without bile, lipid digestion could not happen. This is true. Remember, bile is needed for emulsification. It starts breaking down a large fat droplet into smaller droplets so that pancreatic lipase can go around it, then to make micelles that can be absorbed, and then chylomicrons that can be absorbed into the lacteals. This stimulates motility in the colon, gastroenteric reflex, gastroileal reflex, gastrocolic reflex, gastroesophageal reflex. So gastrocolic reflex, when you're dealing with the large intestine, is the colon. So when food hits the stomach, it's going to have motility in the colon, and it's going to cause most often those mass movements, large movements. I already went over this one. Sorry, it's a repeat. Carbohydrates. My cells are formed during the absorption of which nutrient? Monosaccharides, fatty acids, amino acids, disaccharides. So these are fatty acids. Remember, micelles and chylomicrons are formed. So understanding the process of fat digestion, um, monosaccharides, disaccharides are carbohydrates, and amino acids, they're just going through the cells of the enterocytes of the small intestine. Fatty acids have to go that special way, absorbed in chylomicrons, absorbed into the lacteals, and then go into circulation. So I hope this helped. And if you have any questions, just let me know.